Ash Dieback. It's now becoming quite a familiar tragedy for all of us who are used to going out into the environment. Now it's quite common to see trees that look a lot like this, that have quite bare canopies, and pretty much all across the country. This is just to give you some idea of the variation that exists in the amount of Ash Dieback that we're seeing. This is the Leeds area. You don't really need to be able to see any of the localities here, but hopefully you can see there's a lot of red dots and a lot of orange dots and a lot of green dots. The red dots are trees that are very heavily infected and basically dead. The orange dots are somewhere in the middle of the infection range, and the green dots are healthy. And you can see there's like a huge amount of variation even within quite small areas. And some of this is likely genetic, some of it may be environmental. But what we can say now is that you don't have to go very far to find quite badly infected trees, but you may also be able to find quite happy trees very close by them. Unfortunately, the predictions now look like Ash Dieback may well cost the UK economy around £15 billion over a century. So this is a really important problem for the UK. If you're not familiar with what causes Ash Dieback disease, so this is mostly a problem for our European ash trees, so Fraxinus excelsior. And it's caused by a particular type of fungus, an ascomycete fungus, which is named Hymenopsyphus fraxineus, and I'll be saying that as few times as possible. It's otherwise known as cholera. And it has two different phases to its life cycle, like many of these fungi do. It has an asexual phase, which you can see cultured up here. It looks a little bit like bread mold, really, or something that would be growing on your cheese. This particular fungus is the one that's growing inside the trees, the living trees. The other stage, called the teleomorph, or the sexual reproductive stage, looks a bit more familiar to most of us who like to dig around under ash trees in the summer. These little mushroom bodies are the sexual phase. These are usually popping out of tissue, ash tissue that you find on the woodland floor. And in fact, if you have a look in the summer, you'll find hundreds of these probably under infected trees. So this particular fungus, it has quite a simple life cycle. It's got spores being released in the summer, and these spores can usually travel 1 kilometre or more, but some of them have travelled many tens of kilometres. So you can see that the disease can spread quite quickly with just even a few spores travelling that kind of distance. So these spores are released in the summer, they travel on the wind, and on occasion they will meet another ash tree. And when they do, the spores stick to the leaf, and often this is the first kind of symptom that you see, which is this initial wilting. So what's probably happened with this leaf is that a spore has landed somewhere on the tip of this leaf. And then the fungus starts to grow into the leaf tissue, and it grows through the vascular tissue of the leaf. So as it does that, it starts to block that vascular tissue, and water and nutrients can't travel into those tissues any longer. And that's why you see this wilting phenotype. It's also quite handy in that we can see where the leading edge of that disease has got to. So actually through this wilting, I can tell there's a bit of brown tissue here, that the disease has actually progressed that far into the leaf. As it progresses even further, it will reach branches. And here you can see another kind of characteristic feature of Ash Dieback, which is these diamond-shaped scars that you often see on the smaller branches. And once it starts getting into the branch, then it will continue growing. And in fact, even after the leaves have dropped and through the winter, you'll still see progression of the fungus through the living tissue of the tree. So this will continue through the winter. And then in the autumn, as uninfected trees do, they drop their leaves, and those leaves will then spend winter on the woodland floor, and they will produce the next generation of fruiting bodies the following winter. So this is actually the centre of the leaf rachis, and that's the only bit that basically survives the winter. That's all that's left. And you can find lots of these tiny black rachis on the woodland floor if you look hard enough. Sorry. So because you get this cycle of leaves dropping every year, this disease doesn't kill the tree usually, unless it's a very young tree in that first season. So what happens is that you get a progressive dieback of the tree, often from the tips. So this is often where the spores just hit, either the tips or close to the ground, where the spores are actually being released. And you'll see that the dieback often will start at the tips and then eventually end up in the centre of the tree. And as it works its way into these branches, you'll start to see necrosis on the trunk. So you start to see big necrotic lesions forming on the trunk. And that usually is what leads to the death of the tree ultimately. So Ash Dieback has come from Eastern Europe, and we know this largely because of reports from the early 90s in places like Poland, where they recognised that ash trees were dying in great numbers. At this point, nobody knew that this was a fungal pathogen. They didn't really know what was happening. It had likely been there for quite some time already, and was only really noticed when we had a bad year for Ash Dieback and lots of trees declined quickly. But from this map, you can then see that the spread, these dates are not exact because often the disease hasn't been spotted, including in the UK, as soon as it arrived. But this is often when it's been noted. And you can see just from these dates, really, that over 10 years or so, it spread quite effectively west into Western Europe. Interestingly, the exact same fungus is found in Asia, and so we think it originated from Asia. However, in Asia, it's also associated with Asian ash species, such as Fraxinus and Manchurica. But in these species, those ash are actually resistant to Ash Dieback. They don't get Ash Dieback. The fungus itself is actually a saprotroph, so it's feeding off decaying material in the leaf litter, and it doesn't affect the living trees at all. So this is likely has become a problem because it's been introduced to previously unchallenged species of ash. So the European ash has only recently come into contact with it. And from some research that did not come from my research group, there is indications that this has happened on a couple of occasions. So if you look at the genome sequence of the fungus, that's what this is showing here. If you sequence the genomes of all of the different fungal isolates in Europe over here and compare the diversity of different genetic bases that you see in the European collections of the fungus, if you compare that to some from Asia, what we find is that these long branch lengths mean that there's lots more genetic diversity in the Asian samples than the European ones. And what that tells us is that there's probably just been a couple of founders that have actually started the disease in Europe that have come from Japan. But this also tells us that this could also be a future problem. So if there's a lot of strains that are in Asia that have never made it here, they could be more pathogenic, they could be more problematic. So it's still important that we kind of reduce any additional new strains, this particular fungus getting into the EU. So to where I come into this story, in 2012, I'm sure a lot of you remember, Ash Dieback was discovered for the first time in the wider environment in the UK. It was discovered in a woodland in East Anglia called Ashwellthorpe, and it's called Ashwellthorpe because it has lots of ash trees. It was famous for its ash trees. And as it happened at the time, I was working very close to this wood at the John Innes Centre in Norwich. and that's one of the reasons why I now work on Ash Dieback. One of the first things that happened when Ash Dieback was recognised in the wider environment was, for maybe only, I think, the second time, the government held a COBRA meeting for this as a plant health urgent problem. So this COBRA crisis meeting was held very quickly, and several decisions were made as a result of this meeting. The first thing that happened was that it was agreed that we needed to survey the country to find out how widespread Ash Dieback already was, and also to continue that surveillance to see how fast it was spreading. There was also a variety of control and biosecurity measures. Some of you may be familiar with some of that. So there was almost an immediate ban on the movement of ash products, even within the UK. And that probably affected me more than anybody else, because I was going into lots of woodlands and trying to collect samples, and having to I had a license to do that, and all materials had to be destroyed afterwards and burnt. It's not the case anymore, so don't worry about picking up ash anymore. And there was also a long-term strategy put into place, and part of that was the establishment of plant health science officers, and also a bunch of research projects, of which I was one of the first people to start researching Ash Dieback in the country. And of course, on a local scale, we also needed local authorities to be engaged with helping to manage Ash Dieback in the community. So all of this swung into action in 2012. And so why did we need this? Why was it so important? Well, I'm probably preaching to the converted here, but ash trees are incredibly important in our environment. So they make up around 13% of our broadleaf woodland in the UK, which doesn't sound like a huge amount. There's other trees that are more numerous in the environment, but as you can see, it's quite widespread. So this is the map that shows the density of ash across the UK, and you can see it kind of grows all the way across the UK. There's obviously regions like the southeast where it's very, very common. Ecologically, it's quite unique. So it has a very loose canopy. It hosts quite a lot of other species, so it's quite a hard thing to measure this. But people who are cleverer than me managed to measure that there is around 27 invertebrates that are completely reliant on ash and will not live on anything else. And we also host a lot of rare species of things like lichens and mosses. And indeed, there's about 950 species that are associated with ash that aren't completely reliant on it, but that are commonly found associated with it as a species. So it's a really very important species to our ecology and our environment. So where are we now? Well, this is the most recent surveillance map. So this is one of those things that happened in 2012. This is the surveillance that was started back then. There were these 10K grid areas that were set up, and every single one of them is monitored, and is monitored on a yearly basis. And obviously they can't check every ash tree in every square, but they're effectively looking in each square every year to see for signs of Ash Dieback. And then if there's a case, that gets coloured in on this map. So this gives you an idea now of how widespread Ash Dieback is, and you can see from the colours how quickly it's spread. So those first cases are in these light green colours. So these are the 2012 to 2014 cases, and the first case was somewhere around this, where my pointer is now. And you can see in those couple of years, actually, the first surveillance efforts found that it wasn't only present in this part of Norfolk, but it was also present in the southeast and in a few other places as well, going quite far north. So we had these little spots of Ash Dieback actually when that surveillance started all over the country. And what we now think is that it was probably introduced quite a lot of times into the nurseries in the UK, so around the UK, from maybe the early to mid 90s. So it's been here quite a long time actually, and we only noticed it in 2012. But you can also see from this map how quickly it spread. So those kind of, I don't know what that colour is, turquoisey colours, as the deeper the blues go, the more years have passed. But you can see within the first few years, those lighter turquoise colours, the disease spread very rapidly. And that's these wind-blown spores. By this point, there was a complete movement ban of ash. So really the only way that disease was spreading was through these wind-blown spores. So they're very effective at spreading the disease. And for a while, we'd hoped that there would be some areas. I remember at one point we'd hoped Northern Ireland would be spared, not at all. Like islands, we were hoping were going to be spared, and Northern Scotland, you can see we're still filling in gaps. So these are 2024 cases in orange, and you can see some of them are filling in little gaps where previously we didn't see Ash Dieback, even in quite heavily infected areas, actually. But now we're starting to even fill in gaps right in the north of Scotland. Because this has moved from the east of Europe, this gave us an idea about how badly affected the trees were likely to become. And across the rest of Europe, the mortality rates do vary a bit, but they're somewhere between 60 and 80%. So I think it's fairly safe to say the current estimates are that about 80% of trees will eventually succumb to the disease. So far, so depressing. However, there is some hope. So one thing that was also noticed very early was that a small proportion of trees never show symptoms of Ash Dieback, even though they become infected. So these photographs were taken in Denmark. I work quite closely with Denmark, as I'll show you later in the talk. And in Denmark, the disease was discovered about 10 years before it got to the UK. So they had kind of a 10-year head start on us. And I visited this woodland, I think in 2013. So you can see already the Ash Dieback symptoms are quite progressed in this area by then. And in this plantation over here, you can see there are lots of disease symptoms. There's some dead trees back here. This one is very heavily infected, and there's others that are slightly less infected, but you can see that kind of spindly stalks of the branches sticking out. That's kind of a sure sign of Ash Dieback. This photograph on the left was taken on exactly the same spot, about 90 degrees to the left, and there's a tree that looks absolutely untouched. Now, the inoculum levels in this area must have been so high. There was a lot of diseased trees. So it's very unlikely that this tree was just never infected. And in fact, our colleagues in Denmark had confirmed that the leaves that drop from these trees do have the fungus within them. So we know that they become infected. What they also discovered was that the levels of low susceptibility, they're genetically inherited. So the offspring of these trees also carry that low susceptibility to the disease. And that's important for me as a geneticist, because it means that we can look at the genes of these trees and try and link those to disease susceptibility. So one thing I often get asked, or was a topic of discussion certainly at the time, was whether, given that we have about maybe 5% of trees that will never show any symptoms, whether natural selection is enough. Like, if we just leave things be in the environment, will those trees repopulate ash? Any of you who have ash trees growing near your garden will know that ash is very good at creating new ash trees. There'll be lots of saplings that you'll be pulling out all the time. So some very recent research, again, not my research, but some very recent research that came out this year is showing that you can already see these signals of natural selection acting. This is happening in populations that are infected with Ash Dieback, because you see the trees, the young juvenile trees that are susceptible, are killed off very quickly. So one thing we've known for a long time is that young trees, very young saplings, if they get infected and they're susceptible, they'll die within a year. So very quickly, you start to strip out all of those susceptible trees from the population. And so the next generation of ash, the ones that survive through that initial high kind of selective pressure against the disease will increase the resistance of that younger population. So this is natural selection acting in real time and within quite quickly within a generation. So although it's, and this is polygenic, so this is across lots of different genes in the genome, it isn't just a single gene that's providing this, this level of resistance. But there's a few problems with natural selection. The first is that it can act quite slowly. The second is that you need a relatively high amount of that genetic variation, that positive genetic variation in your population for selection to act on. And the problem is that we don't really know if there is enough, and likely there isn't. The kind of moral from the rest of Europe is that the ash populations are not recovering super quickly. So possibly we need to give nature a helping hand. And so if we can actively manage trees as well, so using things like selective breeding, we can accelerate this evolutionary response and hopefully secure the future of ash in our landscape. So again, this is where I come into it. But before going into my research, I always like to sort of start by thinking about what might actually be the responsible mechanism for this reduced susceptibility. So just to put in some analogies from my recent cold, one way that you can avoid dying from a disease is by avoiding it altogether. And the same is true for trees. So a bit like if I'd have worn a mask a week or two ago, I might have avoided this cold, and trees in the same way may be able to avoid infection. We'll come back to that. Oh, doesn't want to let me move on. Think of one quick. No. Oh, there we go. Brilliant. The next one is probably the first one people think about is true genetic resistance. So the analogous thing for us would be if we've had a vaccination, we have the antibodies against a disease. Plant immunity doesn't work in really the same way, but they do have true genetic resistance if they have a gene that allows them to recognize a specific pathogen. So we call these resistance genes. If they have those resistance genes, then the immune system in the plant is able to recognize the pathogen and basically kill the pathogen. That also exists. And the other possibility is something we call genetic tolerance, which is a bit of a grey area. But the way I like to think of it is that genetic tolerance prevents damage. So a bit like if you take paracetamol when you've had the flu, this is helping prevent the damage that's caused either by your immune response or by the pathogen itself. And in the same way, we may have these mechanisms in the trees that are helping them to stay healthy despite becoming infected. So any of these kind of areas may be responsible for this reduced level of susceptibility. Now on to some genetics. So this is obviously where I came in, was thinking about how we were going to start looking at understanding how the genetics is leading to this reduced susceptibility. And I'm sure everybody's familiar with DNA. So DNA is what encodes the proteins. And if you have changes in your genetic code, then you will have different proteins. And that can be one way to alter a phenotype, so to alter your traits. But another important factor is something called gene expression. So gene expression is the level of activity of the gene. And we can measure the level of activity of the gene and the gene sequence using something called transcriptomics. So this is where we're measuring all of the messenger RNA in a cell. So messenger RNA is an intermediate. So you've got your genetic code, your DNA, and that needs to be transcribed into messenger RNA, which is then translated into protein. So it's kind of an intermediate step. But the number of mRNA units that you see correlate with the amount of protein that's being made. So this tells us how active a gene is, but it also tells us the genetic code of that gene. So we can see if there's sequence differences and if there's expression differences. So I was happily working on a new method at the time in Norwich. I was working on a completely different plant, oilseed rape, which has quite complicated genetics. And so we wanted to use an approach that both simplified the genetics, because oilseed rape has a really huge genome, but doesn't have a huge number of genes. So by simplifying it, we can look at just the messenger RNA, just the coding regions of the genome. And we can also look at both the sequence and expression variation. So I was developing this method, which is called Associative Transcriptomics at the time in oilseed rape. And what this relies on is having a diverse panel of, well, in the case of Ash Dieback, trees. So by diverse, I mean hopefully genetically diverse, so not related to each other, but also phenotypically diverse. So in the case of Ash Dieback, we needed to have trees with low susceptibility, trees with high susceptibility, and trees somewhere in the middle. And having then identified the variation in the gene sequences and expression, really all we're doing is correlating that genetic variation with the trait variation, so Ash Dieback susceptibility. And then this helps us identify regions of the genome that are linked to that trait. So this is what these plots show. They're called Manhattan plots because they look a little bit like the Manhattan skyline. But basically, this is oilseed rape one here. Oilseed rape has 19 chromosomes. And so these are the alternating colours here. And where we see markers, so genetic markers, either genes with variation in their gene expression levels or changes in their sequence variation, where we see them highly correlated with the trait of interest, they're higher on these graphs. So we're looking for points that are high up on these graphs, basically. And we can do this for both the gene sequence and the gene expression levels. Okay, so that's roughly how the method works. It was slightly more complicated getting this to work for Ash. And the reason for that was that at the time we started doing this research, there was no genome sequence for Ash. There had been one for many years for oilseed rape. And also we needed that collection of trees with really good variation in its susceptibility levels. And again, this is where Denmark came in. So the University of Copenhagen, for many years, had been studying their trees and had had the forethought to graft all of these trees. So not only did they know which trees in the wider environment showed low levels of susceptibility or high levels of susceptibility, they'd grafted them and put them into polytunnels and glass houses, which meant that it preserved even the sickest trees which had died years before were still preserved in these protected environments. And that was really important for us because when we're looking at gene expression, one of the problems with working with RNA rather than DNA is that the levels of activity of genes is very variable. So even if you had two twins that were sitting next to each other in the same room, the gene expression levels would likely be very different to each other for any particular gene. This is the environmental effect that we see that affects the levels of expression of your genes. So to try and compensate for that, we actually sample not just a single tree, but we sample multiple grafts of the same tree so that we have this replication. And that helps us kind of account for the environmental variants that are changing the expression levels of the genes. So these are actually 5 different leaves from 5 different grafts of one tree. The other thing that they'd done in Denmark is, as well as having grafts that were protected in polytunnels, they'd also taken another set of grafts and put those into field trials. So where they're all growing in the same field, essentially, all have the same disease pressure. So you can really see the effect of the genotype of those trees on the level of infection or the level of disease that those trees have. So they provided us with the samples that we needed. So we grind these down and extract the RNA. And they also provided us with a fantastically rigorous set of trait data so that we could understand the susceptibility levels of the trees. And then the next problem was that once you've sequenced all this RNA, that's fine. But to make sense of it, you do need a genome sequence as your kind of something to compare all of those bits of sequence to. So some other collaborators at Queen Mary University of London, luckily, were working on the Ash genome sequence. So we were able to collaborate with them and use a very early version of their Ash genome. So there's still problems with this Ash genome in that we had no order of any of the genes. We didn't know where they were in the genome. So our Manhattan plots did not really look like a Manhattan skyline at all. It was just a universe of stars. And then my part of this was to take all of that information and do a bunch of statistics basically on this to try and understand which of those genes are linked to the disease susceptibility. And this is the results. It's a bit underwhelming actually, but this is actually we now do have a genome order. So in the 10 years since the first genome was out, we've managed to put together the order of the genes in the genome. It still needs a lot of work, this genome, and new versions are coming along all the time. But this now allows me to show you where all of those markers that are important are in the genome. You can see they're spread all across the genome. And there's quite a lot of them. So there's about 130 or something on this graph. They're all the ones in black, basically, are the ones that are interesting to us. And this dotted line up here is a very high bar for the level of significance. So only one of them passes this massive high bar, but there's 2 or 3 that are really interesting markers. They have very high correlation when we look at these markers with the disease susceptibility. And so probably when I was starting to do this work, I was expecting that these genes, when I looked at them, would definitely be these resistance genes, right? They've got to be controlling genetic resistance. I've since learned that I'm always wrong when I try and guess what these genes are going to be. They were not resistance genes at all. So they all belong, so actually quite a lot of these black lines, these black dots here, belong to the same family of a particular group of genes called the MADS-box genes. It doesn't matter what that stands for, but these genes are not kind of genes that are directly involved in immunity. They're actually genes that control the expression or the activity of other genes. And this is interesting because hopefully this means they can have quite a large effect. So by changing just one of these genes, that might be affecting the expression of many other genes. So this is interesting to us. However, MADS-box genes are not really known for their links to stress. So they do have some very vague links to stress, or 1 or 2 of them do. But more generally, they're known as genes that control development timing. So classically, lots of these genes are involved in the flowering pathway in plants, annual plants like Arabidopsis thaliana. They're very well characterized. They're some of the best known genes in Arabidopsis. In trees, of course, we're not working with an annual plant. We're working with a perennial, long-lived species. So they're probably involved in the timing of other developmental cues. So we know in tomatoes, some of them are involved in the production of abscission zones. So that's these little zones where you get kind of pinching of the tissue, often around things like your fruit. So for tomatoes, you get an abscission zone when the fruit is ripe, the abscission zone forms and the fruit drops. So these genes are involved in this kind of process. In trees, they're probably also involved in things like the timing of when the leaves come out in spring or when the buds are set. So it's a little bit confusing, but we saw a lot of these genes. And so we hoped that this was an interesting result. And actually, some of the work that was going on again with our collaborators in Denmark gave us some clues about what might be going on. So in a completely separate analysis, they'd found that the timing of senescence, so when the leaves start to turn brown in the autumn and die, this timing of how early this happened was also correlated with the amount of disease damage that you saw. So this is slightly misleading, this graph, but basically they found that if you drop your leaves earlier in autumn, then you're less likely to be susceptible to Ash Dieback. So this was quite interesting because this was another link somehow to development with this disease. And so what our theory is now is that having good timing might be helping these trees to actually survive the Ash Dieback disease. This is only one possible mechanism for what might be happening in these trees. Those of you that are familiar with walking around woodlands and spending a lot of time staring at ash trees, which I have to admit I didn't do 10 years ago, but do a lot of now, you'll notice that ash is quite variable in the timing that it flushes and that it drops its leaves. So you will pass a few trees that flush weeks earlier than others in a very small area. So most of them are flushing somewhere April, May time, maybe. It's also quite dependent on the season and the particular year. And they'll generally be dropping their leaves in some sort of 4 to 6 week period across October and November. Now, why this is important is that the fungus is only really peaking in the summer. So you can find it in May. I struggle to find it really in May, or find these fruiting bodies at least in May, and September is quite late, but there will be a few spores flying around still. The vast majority of them are flying around between June and August. And so what we think some possible routes to resistance might be is that if you flush your leaves earlier, one thing we do know is that there's a trade-off in plants between growth and resistance. So plants can either put energy into growing or they can put energy into resistance, but they can't really do both at the same time. So if you've already flushed your leaves and your leaves are fully grown and mature, then possibly you can put more energy into fighting the disease. So if you flush your leaves earlier, you may be better prepared for fighting the disease. The other alternative is that, or possibly both of these are true, that if you shed your leaves earlier, that the fungus itself is restricted just to the leaf that's going to drop anyway, and that it hasn't had time to progress into the living tissue of the branch. So again, if you shed your leaves earlier, which certainly agrees with the work done in Denmark, that you will also be able to put out a fresh canopy the following year like nothing ever happened, and you'll become reinfected, but so long as you keep dropping your leaves early, hopefully you won't become diseased. So those are that's possibly what's happening. But as I said, a key, this is kind of interesting biology, but a key target for us was to see if we could use these markers to actually predict disease in other trees, because we need to kind of find, or we needed to at the time, find lots of tolerant trees or resistant trees, depending on what these genes are doing. So those top 3 genes that I showed you in that Manhattan plot, we only took 3 of them. And we wanted to see if we could use those genes to predict the level of disease susceptibility in another panel of trees that we'd never looked at previously. And it was a relatively small scale experiment. We only had 66 trees from Denmark. It's quite hard to find really reliably phenotyped trees. Certainly it was in 2013 when we were doing this, because in the UK, for example, we had no idea which trees were tolerant and which ones were susceptible. So we had a relatively small panel, but what we found was that we were pretty good at identifying trees that were going to be really resistant. So this graph shows the predicted level of damage that was based on just those 3, the levels of gene expression in those 3 different genes, and then the observed damage across 2 years in Denmark. And I guess what you can take from this is that the trees that we predicted would have quite bad damage scores, we weren't very good at predicting those, really. And that's probably because there's a large environmental component as to how badly affected a tree is going to be if it's susceptible. But also there's lots of trees down here that we predicted would be quite badly affected were actually pretty healthy. And it could be that we've only identified one mechanism and there's other things going on. In fact, I'm sure that's the case because we're not lucky enough to have solved Ash Dieback with 3 genes. However, what we did find was that a subset of trees that we said or we thought would be very resistant were even more resistant than we expected. So if we thought things would have roughly 30% damage or lower to their canopy, we often saw that they were much healthier than we'd predicted even. So it seems we were able, or these genes are pretty reliable actually at allowing us to find tolerant trees in the environment. So this was great news for us. We published all this work. But the next thing was then to start using these markers. So over the years, these genetic markers have been used quite a few times to try and identify less susceptible trees at different scales. So this one over here, this is a study in Ashwell-Thorpe Wood, where that first case was found in Norfolk. And similar colour coding, as you might imagine. All the red dots and orange dots are trees that we predicted would be faring quite badly under Ash Dieback. And the green and yellow ones are slightly better. And this more or less follows what happened in Ashwell-Thorpe. So it has been very badly affected by Ash Dieback now. But there were a few high points in here. So one tree right in the middle we predicted would be the best one. And for some reason, just because she was a particularly attractive tree, we gave her a name at the time. We didn't know how susceptible she was. She ended up getting called Betty as a nickname. And of course, that hit the news. So Betty is still going. She's still alive. And so, yeah, there's lots of them that aren't. So we also looked nationally. So again, this work was published a few years ago now. So initially, these were the trees. These are the 66 trees we used in our Danish panel to see if we could predict the Ash Dieback. And you'll see there's lots of orange and brown, which are pretty bad predictions, and relatively few green points here. But you'll see the case in the UK is much different. So these are trees that were sampled and again grafted and put into plantations in the UK. This is us sampling them. And you can see that the outlook is much better. There's many more green points. And so we hope this is going to remain the case, that the UK perhaps has a better diversity of ash, and so that there may well be natural selection might act a bit better in the UK than it would do in Denmark. So hopefully, we're not quite sure if that's the case yet, but hopefully it is. And then the markers were also used towards some of the projects like the Living Ash project, which is still going and is designed to find as many tolerant trees as possible. And they're now in these kind of breeding projects. So that was kind of all of our work that was published until a few years ago. Since then, we've refined some of this work. One thing we found is that the markers are not infallible. It turns out that tree size really matters. So all of these markers were discovered from mature trees, but very specific kind of sizes of trees, because the nature of trying to get tissue from trees is that if they're too big, you need a shotgun basically to get hold of tissue, or somebody who's willing to climb trees. So the trees were a very specific age. And you can see that now this latest work, this was actually based on some work that was done in the UK, in UK woodlands. And the markers work really well, actually better than they did in Danish trees, when we look at trees of a certain size. So these trees were all sampled when they were about 25 to 31 centimetres in diameter. So they're relatively kind of middle-aged, not very old trees, I guess. And so we explain about 29% of the variation in disease susceptibility when we look at trees that size. However, if we look at more mature trees, so 40, 50 centimetres, what we find is that our markers don't really work anymore. Now, this is interesting. It's obviously a problem if we want to sample older trees. It's important that we know how well these markers work. But it's interesting because it maybe tells us something about plant immunity, and that perhaps older trees that are still surviving the disease are doing it in a different way to our younger trees. There's lots we still don't understand and lots more research that we need to do. And then some other research that we're doing at the moment is about whether there's other mechanisms, so genetically speaking. And one thing we're interested in is whether ash trees might be able to kind of remember being infected. Obviously, they're long-lived trees, so within a generation, can they remember being infected, and can they mount a stronger response in the following year? And one mechanism that you can do this is using something called epigenetics. So there are chemical modifications that can be induced by stress, and these modify the genetic bases, the DNA bases, but they don't alter the genetic code. So they're not changing the sequence of the genes. What they are doing is these chemical modifications can alter the amount of expression activity of these genes. And this is one mechanism where plants can prime themselves to better respond. So it acts a bit like a memory. These marks on the genes can last a certain amount of time. It's a bit of an argument about how long it can last, but probably several years at least. So this might mean that trees can prime themselves for the next infection and respond more strongly. So a bit of preliminary research that we were doing was looking at these kind of genetic modifications and seeing, we looked at a susceptible tree, which you can see in orange, and compared this to a resistant tree in green. And unlike when we did our first kind of analysis, what we found was that where we see big differences in these epigenetic marks, these are in resistance genes. And one thing that if you have resistance genes in your genome, you'll have lots of them. So trees will be facing all sorts of different pathogens, and we'll have these kind of genes against all sorts of different threats. You can't have them all switched on all at once, because that will have a kind of metabolic, a negative metabolic kind of trade-off for the plant. So you need to be able to switch them on and off when you need them, and epigenetics is one way of doing that. So the fact that we're seeing different epigenetic signatures in resistant and susceptible trees perhaps tells us that there may be a role for this, and trees might have a stress memory that allows them to kind of fight the disease year on year. So hopefully, having had quite a depressing start to the talk, things hopefully are giving you a bit more of a positive outlook for ash. So hopefully with a combination of natural selection on the tolerance or resistance that we know already exists in the population, coupled with careful management, so not just taking out every tree that has early signs of Ash Dieback, leaving those in the environment as long as possible so they can pass on their genes, and selective breeding where we can actually find tolerant trees and try and produce new ones that are resistant to the disease that can be then placed into the environment to replace trees that have died. All of this will hopefully support the recovery of ash in the UK. However, we do need to also be mindful of the new new threats to ash. And unfortunately, there is another one on the horizon. It's called the emerald ash borer. It's a rather beautiful beetle, actually. The beetle itself is beautiful. The larvae are really not. And so what this beetle does is it feeds on the leaves of the trees, but then it also likes to lay its eggs in the bark. And once those eggs hatch, the larvae like to burrow around, chomping through the living tissue under the bark. And the result of that is they essentially kill the tree. So they're chomping through all of that vascular tissue. And so they can kill a tree very, very quickly within a year or two. And this has been a huge problem in the US. So this graph shows the distribution of ash in green. So these are different species of ash to the ones we have here. These are white and green ash, which are predominant in the US. And the red and red points here are cases of emerald ash borer. This has spread relatively quickly as well. So you can see now it's around almost half of the distribution of ash in the US has now got emerald ash borer, and it's incredibly destructive. Unfortunately, it is on its way. So again, it's an Asian beetle. It doesn't cause any harm in Asia at all. But again, similar situation to Ash Dieback. When you introduce it to unchallenged species, you see the trees can't cope. So it has been found in Russia and Eastern Europe, and it's spreading slowly. So I won't go into too much detail of this graph for time, but basically the different colours are showing its spread. So from orange through to the blues and the purples, it is moving west from Russia into Ukraine and also kind of into Kazakhstan and places like that. So we also obviously need to be very careful about this. And at the moment, there is a movement ban on ash from outside of the EU. And if we start to see emerald ash borer making its way into the EU, I have no doubt that we will have another movement ban to prevent the beetle getting in. The good news is the beetle is moving much more slowly than the fungus was, and we know it's coming. So research is already ongoing to see how we can prepare for possible cases of emerald ash borer or produce trees that are more resistant to emerald ash borer. We think European ash, I should say this is the very edge of the distribution of European ash here. So we think European ash is less susceptible than American ash species. Unfortunately, the beetle really likes trees that are already stressed, and we have a lot of those now. So we do need to be careful. But as I said, there's lots of surveillance now going on, and we've learned a lot of lessons from the Ash Dieback epidemic that hopefully will mean that emerald ash borer does not become the problem that it has in the US.